My name is Georgios Kotsakis. I'm a professor of oral biology and the assistant dean for clinical research at Rutgers School of Dental Medicine, and I've been with Rutgers for a little bit over 2 years now. We work on translational, periodontal, and peri-implant research. A particular area that has attracted our attention and we have significant funding on is peri-implantitis. This is really destructive loss of jawbone around dental implants. Over 1 million Americans receive dental implants every year to improve their mastication, to improve how they speak, their aesthetics, and really replace the teeth that they've lost. But unfortunately, 1 out of 4 of them gets a destructive disease that feels a lot like osteomyelitis, where they lose a lot of jawbone. There's pus coming out, there's all the signs of infection. They have to have that implant treated very, very promptly to maintain the structure of their jawbone, of course, right? And their ability to eat and chew food. And unfortunately, we're not successful at all in managing that right now. Or I should say, we were not until recently, where our study really has doubled the success rates of dental implant treatment. We're working on 2 directions. One is actually medication that would block the inflammatory pathways that are involved in causing this disease. That's very specifically related to the metal that the implant is made out of. And the alternative route would be prevention devices. New treatments that are based on novel devices that can clean those implants, maintain them healthy, and maintain both plaque-free implants, but also implants that resist corrosion in the mouth. The reason why we've been working on over a decade now on this treatment is that it really requires combination of human investigation, understanding what really happens in the human tissues, in the human body, and at the same time, the capacity to have a full-on pharmaceutical level lab to be able to investigate down a mechanism. And the way we do things here at Rutgers is highly innovative. We do what we call translational research, which means that we're physically located just a few feet from the dental clinics. So we can go in the dental clinics, collect the tissue, collect the implants, and run them over to the lab right away and get down to single cell resolution. We can either pick up single cells of the human tissue, like the cells that compose your skin layer, for instance. We call them epithelial cells. Or we can get bacterial cells and grow them in the lab and then do downstream assays that maintain that translational relevance, that meaningful translation to human. And it's really outstanding to have this capacity. And as you can see, in addition to the proximity of the clinics, we have this beautiful open lab concept. So you see here, bay after bay after bay has cutting-edge shared equipment. But the most important thing is that all labs share this open space, and in a way there's cross-pollination. So people on the one hand who work on immunology on the human tissues interact with the people on the other end that work on bacteriology, and we eventually can do high interdisciplinary research, very high level. Back in the day, it used to be that you would come up with ideas of what might be relevant and study them in the lab, and then eventually, years and years later, you would go to animals and eventually go to the human. And now we reverse engineer this. We do what we call human-centered research. So having that physical proximity here at Rutgers really enables us to be on the cutting edge and start from the human, understand really the process that's going wrong, and go down to the lab and break it down to its smallest possible components. If you look at our labs, you will see we have major equipment on 3 different disciplines to be able to do that work. On the one hand, we have people who are biomedical engineers and work on metal corrosion, right, and surface characterization, and we have all this equipment. We also have core equipment for bacteriology, microbiology, growing bacteria, and we have experts in that domain. And we also have what is called tissue culture labs, which you mostly see behind these closed doors on the side because they have to be very sterile and they have to have a certain level of safety. And we grow human cells in there, and we see how they interact with either bacteria or the implant itself. So we have to bring all this equipment and mesh it together. We have to bring the people with expertise. And the open lab is the most successful way of doing that while maintaining that cross-pollination that we talked about between the teams. What brought me to Rutgers School of Dental Medicine was that the leadership really believes in research. You know, oftentimes in institutions we say that, yeah, we do, teaching, of course, which is the primary mission. We do service to the community and we do some research, but sometimes it's just not as supported, and as a result, it doesn't become very meaningful. Here at Rutgers, when we say we do research, we really are leading clinical research in our domain nationally and internationally. So that's what brought me here. But I will say, as I was being recruited, when they opened the door, And I entered the labs and I started looking to my left and see bays and people talking to each other as far as the eye could see. That really sealed the deal for me. The good news is that our research was so promising that we were able to work with the Rutgers Tech Fund and get a provisional patent on a possible new therapy. So this is very, very exciting and very cutting edge. And now we're talking to stakeholders and focus groups to understand how that translation will play out. And on the other hand, we've had visits from pharmaceutical companies that work on medication on the other route of treating this disease that we were talking about. And I will say that during their visits, when they see our lab capacity and their potential, they're very excited to collaborate with us. So I would say hopefully in a horizon of the next 5 years or so, we're going to be able to do real human translation, real application of these new treatments in humans.